Hello, my name is Heethoff from the BATS lab at the University of Tubingen, and in this presentation I want to talk about non-invasive three-dimensional visualization of the internal organization of micro-arthropods using synchrotron X-ray-tomography with a sub-micron resolution. The model system that we're working with is the Orbitid-mite Archaeocetus longicitosus, a microchelicerate arthropod of 800 microns to 1 millimeter in body size. This is a picture taken from our lab culture where you can see the animals feeding on algae. To study the internal organization of Archaeocetus, we use synchrotron X-ray radiation to produce tomographic voxel data at the ESRF in Grenoble. Here you see a picture of the ESRF with a large storage ring, and in the upper right corner you see the experimental hall of ID19, which is located outside the ring. For the measurements, samples were critically point-right and mounted on plastic pins using superglue. The experiments were conducted at 20.5 keV and 1500 projections were taken for the reconstructions. Here you see the camera with a cooled freelance CCD chip, 14-bit dynamic range, and 4 megapixels. The scintillator, shown at the bottom of this picture, translates X-rays to visible light. This is a close-up on the sample holder and the rotation table. The sample is mounted on a goniometer head to enable a proper orientation of the sample in the beam. A detailed description of the experimental setup is given in our 2007 paper in the Journal of Microscopy. In this presentation,